This workshop is on hacking the code of life. So it is kind of just a brief introduction about biotechnology and computational biology. And this will be geared kind of more towards people who have kind of an engineering or coding skill set, but are looking to apply those skills and those technical knowledge to an area that is very cool, so biology, or other fields within biology, so like neuroscience or things like protein folding and stuff like that, which we'll discuss. So just to get started, introducing myself, my name is Jennifer. I'm currently a fourth year biomedical engineering student here at Waterloo. I am a computational biology and neuroscience researcher. I've done a lot of research in the past in these two fields. And I'm also a managing director of Nucleate Dojo, which is a nonprofit that helps train undergrads to be successful in the biotech industry. And for hobbies, I also really enjoy reading, writing, and hiking. And you can connect with me on my social medias down there. Just a brief agenda of what I'll talk about today. So I'll start with why I'm in biotech and my story of what keeps me going and what keeps me motivated. I'll go into a high-level overview of what biotech is and what the emerging fields are within the field. And then I'll talk a little bit about some computational biology technical examples. So some specific concrete examples of how coding can be applied to solving biological problems. And then I'll end off with kind of a call to action for how you can get involved in biotech and why you should care about this exciting field. So I think a lot of the people that I look up to in biotech and in science have a very compelling story of why they are in the field. And I think science and biology is very hard and slow compared to software and coding, where a lot of the results are pretty immediate. With science, that takes a longer time, and discovery can be very arduous and slow. And so there always kind of needs to be this underlying motivation of why you're pursuing what you're doing in biotech. And I'm going to share 3 of the stories that keep me going and I hold close to my heart. So this is a picture of me and my dad when I was graduating high school. This figure, my dad is a big figure in my life that motivates me on a daily basis. We have a really close relationship growing up, but I think something that I've had to deal with growing up is that he is actually diagnosed with post-polio syndrome, which is a condition that results from the aftermath of getting polio when you are really young. And so when he was around 1 or 2, he got contracted polio. And then as a result, one of his legs is paralyzed and not able to function as a normal human being. And so growing up, I kind of saw him not being able to engage in regular physical activity, such as running or walking long distances or climbing long flights of stairs. And so seeing his quality of life kind of not to the fullest extent of a regular human being was something that got me really interested in science, and especially kind of the regenerative medicine and neuroscience field. And so that was kind of my first spark into science. And then in high school, I was volunteering at a physiotherapy and neuroplasticity clinic in BC, which is where I'm from. I had the privilege of meeting this amazing individual. His name is Trevor Green. He's actually a war veteran that fought in the Afghanistan war a couple of years, or probably 10, 15 years ago. But while he was in Afghanistan, actually, he got axe blow from a Taliban soldier who kind of attacked him from behind. And so as a result, he had severe traumatic brain injury. And so he's got sent to a hospital in Europe, in Germany, I believe. And he was kind of in a coma for a couple of months. And all the doctors were like, yeah, we probably cannot like bring you back to your original state. You probably won't be able to communicate, walk, or even like talk to your loved ones. And obviously that's like one of the most like heartbreaking things to hear, as like anyone, I would say. But I think this clinic has been working with him specifically and a bunch of other individuals to figure out if there's any technologies out there that can help him relearn the ability to walk again. And they have been pretty successful, and like over like a couple years, 10 years, I would say, they have been able to get him back into a state where he could walk with a walker now. So that was also very fascinating and inspiring to me. And then last but not least, that's a photo of my grandfather, who unfortunately passed away this past summer due to end-stage Parkinson's. He was struggling with this condition for like 2 or 3 years now, but over this past summer, it got very debilitating, and he had to go to the hospital because a lot of the like fluids that he was consuming, they were going down the wrong system. And so at the end of his life, he wasn't able to, communicate with anyone, even like eat by himself or perform like daily functions by himself. And so that kind of story also really grounded me in why I am pursuing what I'm doing now, and kind of bring all these 3 stories together. Sorry, I don't know how to get rid of that block of text thing, but yeah, so it got me really inspired to pursue kind of neuroscience, understanding the brain, disorders of the brain, mind, and the nervous system, because they have a huge power to fundamentally change how we interact with and experience the world, and even alienate and segregate us from society. All these 3 people that I've just mentioned have had some sort of disorder of their brain or their nervous system, and as a result, like, couldn't function as a regular human being. And so, yeah, that's kind of why I was super fascinated by this field originally. And so this kind of brings me to my, I guess, underlying motivation. I'm marveled by how these interacting systems in the brain and the mind give rise to our complex behaviors. And I'm really motivated and really excited to understand and put order to it. So that is kind of where I'm coming from here and where my story starts. And I think it's always cool to see like how someone has kind of walked through their career journey. And so I'm going to be kind of sharing some areas of biotech neuroscience that I have explored already through internships, through co-ops, and give concrete examples of some like fascinating problems that you could work on as well in biotech. So the first thing, I mentioned this already in my story, but I was working in a neuroplasticity clinic in Surrey, BC, where I'm from, looking specifically into neurostimulation and neurotech that helps people regain the ability to walk again after kind of a traumatic brain injury or any type of neurodegenerative disorder. This was super cool. I learned a lot about kind of clinical research, medical devices. You can see me there with like an EEG cap, and on the top, kind of an exoskeleton that helps people rehabilitate from walking and gait disorders. And then I moved on to kind of looking into big data and using computational approaches to analyze large sets of data. Specifically, I was looking into brain memory here at UBC. I'll talk about this a bit later in the comp bio examples, but this is kind of a cool application of how you can use computer science and big data to understand how the brain works. Actually, last fall, I was at a startup in Boston that looked into mRNA therapeutics and how to optimize those for cancer and different disorders. I was also leveraging a lot of computational tools to optimize these and like discover new drugs that can be applied to cancer and rare diseases. So just another cool example. And last but not least, this past summer I was in Switzerland at EPFL looking into spinal cord injury research. There is this like project that my lab also came out with this past summer called the brain-spine interface. that basically allows a paralyzed man to control this like simulator device with just his like brain signals. And so it's a combination of like a brain computer interface and a neurosimulator device that helps him regain the ability to walk after spinal cord injury. I was working on this project, but I was specifically looking at more on the molecular and like cellular side of spinal cord injury, which cells are responsible for spinal cord regeneration. So that was kind of like, so far in my career journey within biotech, some concrete examples of things you can work on and might be interesting to look into. So now that you have some like examples of labs, companies, problem spaces that are within the biotech space, I'll kind of bring us back to the bigger picture of what biotech even is and why you should care. So biotech is a field where we leverage innovations in biology to create new applications. We also use biology and technology to make discovery and science faster and more efficient. As I mentioned before, a lot of research is pretty slow and arduous because of the current traditional processes with how we do things. So now that we have all these technological and engineering advances, we can apply that into biology and make things a lot more faster. And last but not least, scaling biology innovation. So if you think about like different medicines and a lot of like medical devices, these things are super costly to manufacture and scale to the general population. And so how can we, use engineering and manufacturing and mechanical principles to bring these medicines and therapies to a greater population of people? So I just want to talk about this like concrete example of a therapy that is being developed and highly researched in cancer called CAR T cell therapy. It is essentially what happens is you go to a clinic, you get your blood drawn, and they'll extract cells that are CAR T cells that they then engineer in the lab to make it so that it can attack cancer cells, and then they'll put those cells back into your body so that your body can naturally like fight off these cancer cells. And so I just want to like pose a question. What do you guys think like the price of this therapy is for one individual? It can be like wild guesses. Yeah.
500,000.
That's actually pretty close. Anyone else want to guess? Yeah.
100,000.
Yeah, you guys are actually like sandwiching the number right there. But yeah, it's actually $475,000 for one individual, and it's only for 44% shot at survival, which is like an insane number to begin with. But I just want to point out here that this is a engineering problem, right? The problem here isn't really like the science or that these discoveries aren't like working. Like 40% is not super high, but that is also because a lot of these therapies are very individualized. So you have to extract cells from individuals, and it's not easy to like scale that to like everyone, I guess. So the problem here is that we need more scalable immune cell manufacturing platforms. And I guess the whole kind of, if there's anything that you want to take away from this presentation, I would say it's this slide. Science is accelerating at a really rapid speed right now. We're cracking down on answers to many scientific problems, and each day we're coming up with these like new solutions, but we're lacking a lot of builders and engineers and designers like you guys that can kind of scale these solutions to a greater population and make these discoveries or bring these discoveries from the lab bench to the real world and to patients and to people who need it the most. So yeah, we need more people like you, and we need more people with technical skill sets and like engineering design backgrounds to to join biotech and to innovate within biotech. Yeah, so I'm going to go into more concrete examples of emerging fields and what biotech can solve. So there's 4 emerging fields that I kind of took out here. Not all of them, but I wanted to highlight some that were engineering focused. So computational biology, which I'll go more in depth, bioelectronics, nanotechnology, and neuroscience/neurotech. I'll go into these more in depth as I go on, but I'm going to start out with computational biology because I think a lot of the people in here have at least some sort of coding background or like engineering background, and this field I think is kind of right up your alley and you might be most interested in exploring. So it's essentially applying computational methods to analyze large collections of biological data. If you think about like a lot of biological processes, like how the brain works, how drugs may like act on different cells in your body, a lot of these interactions and processes are very like qualitative, right? Like it's really hard to say like, oh, this brain interaction like is X, like I guess number of synapses or connections to this brain or this part of the brain. But I think computational biology gives us a framework and a tool to represent very chaotic and very complex biological data in a quantitative way and allows us to decode patterns within the body and the human body that we might not have been able to do in the past. And so I'm going to go over 3 main, I guess, like topics within comp bio that I think are the most like hot and like trending right now. So connectomics, transcriptomics, and RNA/protein folding. I'll begin with connectomics, which I think is probably one of the most hardest problems in biology and in biotech. So the human connectome is a comprehensive map of the neural connections in the brain. So if you think about like your brain and how many different like interacting components it's essentially creating a map of that and trying to figure out like what are the dynamics, what are the networks within the brain. And why this is important, because to figure out the network structure and dynamics of the brain can allow us to predict the brain's activity and function. So if we're trying to investigate, let's say, a certain disease or disorder, we can kind of look into this simulation or this model and predict like, oh, if this part of the brain like stops working, then this is what happens and this is what the brain will do. Instead of like using a bunch of experimental approaches, which can be very slow and very costly. So just to give some background, the brain is made up of a network of a bunch of neurons, which are synapses, which are communicated between neurons, and action potentials. And so I want to pose a question again. Does anyone have an idea of how this structure or this network can be modeled computationally? Like if there's any specific like data type or anything that you think? Yeah, in the back. Yeah, that's a good guess. Yeah. Yeah, so we have a graph and a vector, and actually like both those things are correct. So a lot of connectomics right now is using directed graphs to model the brain's neurons and synaptic connections. So as you can see here, the neurons can be seen as nodes, and then the synaptic connections can be seen as the edges, and then we can model different like interactions of the brain using like the graph network theory. And so I guess to make things a little bit more technical, as I mentioned, like you can kind of use the nodes to represent neurons, in which each neuron is a point in the 3D space. And then connections between neurons can be represented as, a neuron pair, and then the number of contacts those two neurons have, and that's kind of like the weight of the directed graph. And then usually, this is a very high dimensional like data type, like a vector. We don't want to usually like use that directly to analyze a system. So usually would create something called an adjacency matrix, which is common in like graph theory to represent graph networks in a more simplified manner. And then once we have these data represented, we will use something called motif embeddings. So if you look down here, these are like different combinations of 3 neurons that have like different connections to each other. So it can either be like this connected to this, and then this connected to this, or this connected to this connected to this. So there's like a total of 13 different combinations. And what we want to do is to see like if there's any patterns between different regions of the brain that have like, maybe like the left part of the brain is more highly expressed with like number 1, and then the other part of the brain is more highly expressed with like 13. And so oftentimes like different parts of the brain have, more or like less of a certain motif. And then then from there we can see how those interactions can manifest into different types of function and different types of behavior that is as a result of that circuitry. Any questions so far, by the way? I don't think I've stopped to ask yet. Okay, I'm going to continue. So I guess just an application to this, there's actually like a pretty big effort right now, or yeah, it was a pretty big effort at EPFL in Switzerland called the Blue Brain Project, which was an effort to digitally reconstruct the mouse brain in 3D. And so there was like 30,000 neurons or more, I don't remember, that was being simulated, and they were trying to run this on like a bunch of quantum computers. I think this project, when I was in EPFL this past summer, I think it was actually shut down because it didn't work. So this kind of shows that this is a very, very hard problem, requires a lot of like, computer science principles, engineering, even like quantum computing. So if this is something that interests you, don't let this like discourage you, even though this has been worked on for a couple years. But yeah, there's some other projects that are up and coming that have been trying to work on it. Yeah, good question.
Does this work for like species with even smaller brains that you would expect?
Yeah, I think there was success with modeling a zebrafish brain, just because it's a lot like less neurons and not as complex, obviously, as a mouse. But yeah, they're trying to kind of move up the species, so like zebrafish, mice, and then like ultimately like humans. So yeah, I'm not sure like why the project failed, but yeah. But yeah, just also to point out, the reason why this is so important and why people, like so many people want to do this is that like mapping the brain helps us understand like what can go wrong and why. As I mentioned in the first slide, like a lot of these experiments on mice and on like monkeys and like on humans, they're very costly and very slow. So if you're able to like simulate this on a computer, let's say, then it really like cuts down on those costs and resources. Yeah, so I'm going to move on to transcriptomics, which is kind of my personal favorite just because I've worked in this space for a couple of terms already. So transcriptomics is an analysis method that allows us to characterize genomic expression of single cells. I know that's like a lot of technical terms, so I'll try to break that down. So in our bodies, we have a bunch of cells that are building blocks of our organs, our bodily functions, like pretty much anything is made up of cells in your body. And there are different types of cells, right? There's skin cells. There are like neurons, which are brain cells. There are like hair cells, which are all really different and they function in different ways. So they have different characteristics. And how are they characterized and how are they different from each other is defined by the genomic expression, their genetic profile. So the skin cells might have a certain type of gene that is like highly expressed. So there's more of a certain gene, whereas a neuron might have a different type of gene that is like more prevalent in that cell. And so you can kind of imagine that like doing this genomic profiling is the basis for understanding a lot of biological markers and like genes that are kind of the basis for like specific diseases and disorders. So if we're able to kind of like map out, let's say your brain, and see like, oh, this gene is responsible for like Alzheimer's, or this gene is responsible for Parkinson's, that's pretty powerful because then we can use that knowledge to design therapeutics and treatments to target those genes. So kind of just background biology of transcriptomics. There's this sequencing method called single cell RNA sequencing that allows us to basically like sequence and pin down individual cells and then extract their gene profile. So how this works usually is we have like a tissue. This can be like a tumor, your like brain, like pretty much anything. And then using this technology, we will like isolate and sequence individual cells. And then using another technology, we will then kind of find the genomic profile of each individual cell. So yeah, this is kind of the workflow. So like once we kind of have the genomic profile of each cell, we can group together cells that have similar genomic profiles. And we kind of define cells with similar genomic profiles as different cell types. So there might be a certain cell that has, you know, this gene highly expressed, and that's kind of responsible for a certain condition. And then another type of cell that's like really low expressing like this gene, then that is not responsible for a certain condition. I'll go into this like more in depth in the next slide. Just wanted to give it a brief overview. But coming back to like the computer science and engineering principles, does anyone have an idea of how we can, you know, like group these cells together and like identify cell types using like purely data science principles? Okay, I'll give you guys a hint. It involves a lot of clustering. Actually, I'll just like show you the answer because I know we're running a bit low on time too. So as you can see in this diagram, I'll kind of explain this a bit later, but this is a result of genomic clustering. So if you think about things like KNN, which is K nearest neighbors, this method essentially takes a large set of data and then is able to like cluster together cells or like features, or sorry, cluster together data that have similar features. And features in this case is like similar gene profiles. And so going from here, from cells and their genomic data, we can create this like matrix essentially with like each cell and like how much expression each gene has in that cell. So it could be 0, which means like there's none of that gene in there, or it can be like a really high number, which means it's really highly expressed. And then we can do something called a linear dimensionality reduction. So this matrix is like super huge. It's usually like tens of thousands of dimensions. So we do something called dimensionality reduction, which compresses the data to a smaller representative data set. And then we can extract like those representative features from that. And then from there, we can do clustering. So we would group together cells that have similar gene profiles. So 0 here has, like all the cells here have similar gene profiles. And then cells here have similar gene profiles. And then we can see like, essentially these are different cell types. So this cell type is different than this one. And then, yeah, it kind of like shows on kind of this graph here, which is done by UMAP, nonlinear dimensionality reduction, to construct this like graph that represents a very high dimensional data set. And so from here, you can essentially try to start to understand what exact features are differentiating these different clusters. So there might be certain like genes that differentiate this cluster with this cluster, and those might be responsible for certain diseases and certain like disorders. And so this is like usually the workflow that people use, scientists use to identify, like gene markers in Alzheimer's, gene markers in like stroke patients, or pretty much anything. It is just pretty much getting like sequencing data, clustering it, and then identifying like cells or genes that are statistically different from each like cluster. Sorry, that's like a lot of technical details. Does anyone have questions about this? Yeah. For a linear one, or?
Yeah.
Yeah, usually it's PCA, which is principal component analysis. I think that's a standard one. And then for nonlinear, it's usually the UMAP, yeah, I think it's usually UMAP. So kind of bringing us back to the higher and like bigger picture of the applications. So what can you actually do with this? Firstly, you can map out clusters onto space. So once you have like what I was showing here with the clusters, you can actually map that back into like a brain tissue to see like exactly where these cell types are like in space, which can be really useful to, target like a specific brain region if you know like that's the cause of a certain disorder. Secondly, you can, as I mentioned, like identify genes that are responsible for disease. I linked a paper here in the slides if anyone's interested in looking into it further. And then the last thing I think is the most interesting, personally. There's a lot of work being done around looking at the gene profiles of kind of like lower level species, so zebrafish and mice, and some other like reptiles, non-mammals. And so if you think about like zebrafish, and there's another mammal called like spiny mouse, these organisms, they can regenerate their spinal cords, but if you think about humans, like we can't do that. And why is that? It's because there are certain genes that we have, or that they have, sorry, that are not like turned on in humans. And so if we're able to like identify those genes that they possess and we don't, then maybe we can like turn those on in humans and like, have the ability to regenerate your spinal cord. Obviously it's not that like simple. It's very nuanced. There's a lot of nuances, but that's kind of the general idea. And if you can see here, like, actually, sorry, this is not super clear, but it's essentially showing like there's a specific type of gene that they identified that is in zebrafish, that without it, the spinal cord cannot regenerate. But with it, after like two weeks, I believe, the spinal cord was able to like regenerate again. So this gene essentially like regulates the function of regeneration, and they're trying to figure out like, can we turn this on in like a human or like a mammal? And so the last technical example I'll go through is RNA folding, which is also a very hot topic right now, or I guess RNA/protein folding, but I'm going to specifically focus on RNA. So RNA is, they are biological molecules that give information for how and which proteins are synthesized. So proteins, to bring us back to definitions, control a lot of bodily functions. So they can pretty much like instruct your body to, pump blood or send signals from your brain to your spinal cord. All these things are governed by proteins. So RNA is very important because they essentially are instructions for how proteins are coded and synthesized. And so figuring out how RNA strands are folding can help us predict how these proteins are functioning, which is, one of the biggest problems and ideas in biology right now. So kind of bring us to the biology side of things. RNA, they are made up of base pairs. So A, U, G, and C. So they're kind of like Legos. You can think of them as like building blocks of a longer strand. And secondary structure is formed when there's kind of like interactions between these base pairs. So it starts off like this, just like a single strand. And then due to like interactions between the AUGC, they can kind of interact and bind to each other to create like a secondary structure. And then something more complex after that is a tertiary structure. So it looks like this. And so this essentially is how the folding works. But before we kind of can predict how the folding works, we need to be able to characterize and represent these like building blocks. Oh, sorry.
So in the secondary structure, what type of chemical forces actually influence the structure? Like it's similar to like protein folding.
Yeah, it's like really similar. It's really similar to protein folding. I believe these might just be hydrogen bonds. Not entirely sure, but these are like stronger forces, like just like covalent bonds, essentially. Within the secondary structure, but I think tertiary structure might be a weaker force, like hydrogen. Yeah. So I just wanted to pose another question. Does anyone know how we can characterize and represent these patterns? So keep in mind that these building blocks are A, U, G, and C. So they're like letters of an alphabet, which should give some hints. Yeah, strings, which can be seen as words or like natural language. So I'll kind of give an answer. A lot of RNA sequences or even DNA proteins are represented as strings of natural language base pairs. And then we can like apply like natural language processing to predict or to extract patterns and to then make predictions. So I have this it's actually from like one of my co-ops, a project I worked on. So I was looking into how we can predict how the RNA binds to specific proteins. So what I did was I took like an RNA sequence, which is just a string of like A, U, G, and C's. And then this is like seen as like a sentence, if you know like NLP. And then we can break these sentences down into words. And so I split these into 6 base pairs each. And then this kind of forms like a vocabulary with a bunch of words. And then using like a natural language processing model, we can feed these words into like I use like a Word2Vec, which is like a NLP model. And then we can extract patterns from there. And so we use these like 6-mers or k-mers to pretty much preserve the sequential and positional information of RNA sequences. So representing the sequence is like one of the, the harder things in this problem, because we need to make sure that we have both the localized features of like where they are, where these base pairs are in the string, but also like how they can bind to like other sequences, and that comes with these words. And so once we have those once you kind of go through the NLP model, it'll give you kind of like a learned embedding of how these different words or these k-mers might interact with each other to bind to each other. Then we can feed that layer into a classifier. And so for me, I use a CNN to make the predictions, but a lot of people have been using other things. Yeah, so that's kind of the basis of how I think most RNA folding, protein folding have been approached in the field. There are a lot of different combinations of classifiers and word-to-vec, NLP models that are being used, but that's kind of one approach. Anyone have questions about this specifically? Yeah.
Is there a specific reason why you use 6?
Honestly, that's one of the hyperparameters that was being tested. I think a lot of people in literature just use 6. Not sure why, honestly, but I also tried with 4 and 3, but I think 4 and 3 are a bit too short to characterize how this specific word will bind to another word in the sequence. So it's kind of just literature standard, I would say. Yeah. Yeah.
How do the labels work? Because I'm assuming you're just, how exactly are you extracting patterns here?
Oh, with the words?
Yeah. How do you know what? Because if you just stick in a string, how do you know what's actually binding to what?
Yeah. So there's usually this is something I probably should have added, but there's kind of a database that has kind of characterizes how different these k-mers can interact with each other. So let's say AAA, they're more likely to interact with UUUU. And so there's this database out there that is able to I guess predict has the percentage of how it binds to each other, if that makes sense. So it just kind of shows or has there's this percentage of possibility that this k-mer will bind to this k-mer, and then that's being used to predict those or extract those patterns. Yeah. Any other questions? Yeah.
I just want to clarify the entire structure. So is this to just classify what kind of RNA it is? Because the NLP side is just getting some semantic information, right? And you have to do a classifier. So then the classifier is the role, you want to classify different.
Yeah. So this problem specifically I was working on was trying to look at something called RNA binding proteins, and which proteins will bind to which RNA. So so let's say we have this input RNA. We want to see which proteins will bind to this. So that was the problem of this. I should have clarified. But I just wanted to also kind of highlight how usually RNA sequences are represented. And this is usually the way people have been doing it. And it's pretty similar for protein folding, I would say, and DNA as well. Any other questions? I think this is the last technical example, so we have some time. Yeah. Sorry, connectome?
Yeah.
Did you say?
Yeah.
I don't think there has been a lot of work being done around that, but I think with the connectome, that's mainly the interactions between neurons, and less so the RNA or like the genomic patterns within it. But that could be really interesting to like include within like a connectome model. Yeah, I think connectomics it's mainly like characterizing the interactions between different neurons and the structure. Okay, I'm gonna move on, but if you have any questions, feel free to put up your hand or come up to me later. Yeah, so just to kind of wrap up that section or that example, there's a lot of applications to this actually, very, very impactful or potentially very impactful. This is not an example, or sorry, this is not an application, it's just something that I wanted to put. It's like an interactive server where you can input like an RNA sequence and it'll show you like a structure of how it looked like. You can kind of play around with that if you want later on. But one of the main kind of applications of this is designing optimal mRNA therapeutics and mRNA vaccines. So as I mentioned, like RNA essentially codes for certain proteins. And so if you're able to kind of optimize, or if you're able to kind of manipulate the sequence in a way that will allow you to code for a specific protein that you want, then that can be very powerful in like designing, you know, certain therapeutics that target certain diseases. So at Strand Therapeutics, where I was working, they actually use like mRNA therapeutics to target cancer. And so their whole like problem, I guess, is to like optimize the RNA sequences so that they can effectively target like cancer cells, let's say, or like kill those off or whatever the mechanism is. Same for vaccines, very similar application. And then the last thing I want to mention is like protein folding, because it's a pretty similar example technically, with how like data is represented with the amino acid sequences. Like those are also like letters and can be represented as like words in NLP. A huge effort that has been like done in the past is AlphaFold. If anyone hasn't heard of it before, it's essentially Google DeepMind's project to fold a bunch of proteins. They have this like huge like model to do that, and there's a example here linked that you can look into to see how they did that, and yeah, run the model there. It just takes some time, by the way, if you want to try. I think it takes a couple hours. This is why I didn't like put in this workshop, but if you want to explore on your own, this link is there. So those are some like 3 technical examples of how you can use like engineering and like a lot of computer science principles to understand biology better, and honestly to like make a lot of new pushes within biological discovery. But I wanted to bring us back into kind of more of the practical examples. And like, I know a lot of you guys are probably interested in like maybe like finding an internship or like working full time at like potentially a biotech industry biotech industry company. So I just listed some of my favorite biotech startups that are doing like stuff like within comp bio. So there's a variety here. There's kind of like using AI to discover drugs for genetic disorders. There's like intelligence platforms to track infectious diseases, and then Strand is where I was working, programming mRNA therapeutics. But there's like a whole lot out there. If you search like one of these, it'll probably show like related companies or like similar companies. But yeah, if you have any like specific like interest in mind, you can come up to me and I can try to think of some companies that are doing those things. But yeah, it's a pretty wide range out there. I also wanted to kind of show you just a brief examples of other non-comp bio fields in biotech. So there's bioelectronics, which is just applying electronics to biological systems. So like there's this like cell rover that operates within like your cells. To be honest, I don't really know what they do, but it's like a very small like robot that like is able to do some sort of like function in your body. There's also like smart bandages that help you heal faster, 25% faster. Nanotechnology is essentially like atomic machines. So there's like Vena Medical, which is actually a Waterloo startup that has the world's smallest camera. It's able to go inside like veins and arteries to help physicians treat like stroke and different diseases. There's these like sunscreen stickers where if you're like UV or if your UV protection has run out from your sunscreen, it'll like turn a specific color to remind you to apply more sunscreen, which prevents like skin cancer. And then a lot more like molecular machines that go inside your body to perform a specific task. And last but not least, there's neurotech, neuroscience. A lot of it right now is like BCIs, brain-controlled interfaces, using your brain signals to control specific things. Like it could be, I don't know, like a car, like pretty much anything, if you can characterize it properly. There's also a lot of like exoskeletons right now being done to help with neurorehabilitation, so helping people, you know, with paralysis, with like gait and balance deficits, like walk again. Yeah, but that's kind of like I wanted to kind of showcase some other fields other than comp bio. So those are some of the, I think, emerging fields right now. So why we need you at biotech? I'm going to end off kind of with call to action. So I think there's a huge like perception that like if you want to be in biotech, you need to be like very knowledgeable about biology and need to have like a PhD in biology. But I think like the only prerequisite you need is to be interested. If you end up like working in a biotech company or a biotech startup, there's going to be like PhD students, there's going to be researchers that are highly knowledgeable about that biological topic. So you don't have to be the one like having all that knowledge. You just need to be the one being able to like translate that engineering and technical skill set to that biological problem, which, yeah, as I showed with the examples, is like not too big of a bridge. Like you just need to be able to like understand like a basic prompt and then see how you can apply principles of computer science into that context. And yeah, there's a million of unsolved problems in biology that I think we can use more hands-on, specifically with people that have like computer science, engineering backgrounds. I linked this like Wikipedia page with like unsolved problems in biology. There's a ton, from like neuroscience to biochemistry to the evolution of life. Like there's a lot. If you want to look at it, there's you can maybe like look into different like topics within the field, how to get involved in biotech and start exploring the field. This is in the hack pack, I believe, but I put some like tutorial, like Google Colab examples, papers that are relevant to the examples I went through. Not going to go over these in detail, but the resources are there. I'd also created this Notion page, which I'll show you right now, that has like pretty much anything you need to know to get started in biotech. There's like biotech podcasts, YouTube videos, courses, books. If you want to go one step further, there's like events and communities you can join. A lot of these are online, so it's pretty low commitment. There's like programs and fellowships you can join if you want to like meet people and actually like get more involved. And I think maybe the most people, like this is probably the most interesting for people here, is to how to get a job within biotech. I would say the biggest hack is to go through these like VC job boards, because a lot of these have like startups that are like biotech focused. So if I like click into like IndieBio, for example, and filter by, or these are all biotech actually. So these all should be biotech companies. You could, you know, email them, you could reach out to their team and see if they have internship roles. But I would say like a big portion of how I find my roles is like through these job boards at VC firms. If you want to like get into research, there's a bunch of other like research programs. When I was at EPFL this summer, I applied to one of these programs, which I think is in here. And then if you're at the very end stage of biotech and you want funding, there's some funding sources here. This is, I think, linked in the hack pack, so feel free to look into that. And also there's a QR code here, which I'll leave up for a bit. But I think that's probably the end of my presentation. I'm going to, I just wanted to do a quick promo of Nuclei Dojo. It's a nonprofit that I'm running. It's essentially a training ground and a community for future biotech leaders. If you're interested in following us, that's our website and our social media. We do a bunch of like programming, have like a biotech hacker house over the summer. We have like a funding program for research. So a lot of cool stuff. But I'll end off with like my social media. That's where you can find me on Twitter, Instagram, and LinkedIn. And yeah, that's pretty much all there is to my workshop. Sorry I didn't leave a lot of room and time for questions. But if you have any questions, I think this is the last workshop in this room. So you can feel free to like come up to me, or I think we can stay in this room for a bit, right? Yeah, so I guess I can take some questions now as well, if anyone has anything. I'll put it back to the QR code. Yeah. I think connectomics is pretty like research based. So a lot of the work being done there is like at research labs, I would say. Like the Blue Brain project, there's also the Human Connectome project, I believe, is still up and running. I think there's a prof here at Waterloo, Chris Eliasmith, that does similar stuff. Yeah, I would say they're mainly like in like research labs. Yeah, at this current stage. Any other questions? Yeah.
If you were in biotech, what was STEM?
If it was in STEM, I'd probably do like that's a good question, actually. I haven't thought about that.
It doesn't have to be exciting.
Does it have to be what?
Exciting.
I don't know. I think I'd be doing like psychology, like something psychology related. I guess it's not really like tech, but yeah. Any other questions? Yeah, in the back. Yeah, that's a good question. I think for high school students, I know a lot of like people get started within like research just because it's like a good exposure to what like the life sciences field is like, while getting like a lot of like hands-on experience. Because yeah, to be honest, I think a lot of these like companies, VCs, like startups, they're less likely to hire high school students just because of like lack of experience. But I think usually like academic labs are pretty open. So I have, I think in this Notion doc still like some opportunities, or sorry, not opportunities, but there's like an email template or something that you can use to reach out to, yeah, that you can use to reach out to like any professors, labs that you're interested in. But yeah, I would say start with like looking at specific like areas and topics in biotech you're interested in, and then seeing if there's any like labs or profs where you live that do that work and reaching out. Yeah. Oh, and there's also a bunch of like, I just thought of this popped into my head, but there's, I think, some like biotech related like science fairs, competitions. There's a Sanofi BioGenius thing or competition, I believe. There's also like the Brain Bee, which is like a neuroscience competition. Yeah, if you like come up to me later, I can think of some other like programs, but I think there's some out there for like high school students specifically. Yeah. Yeah.
building a biotech startup or project idea, you know when you can also get funding?
Yeah, I have some here for funding. I don't know if, I think some of these are biotech specific. So the Delta Fellowship, I believe, is like, oh, I guess they're non-exist anymore. But I think a lot of these VCs that I've also listed in under companies do like early stage funding. These are all like they fund like biotech companies. So these are good targets to have. Yeah, and then these I think are just general like fellowships or funding sources. Yep, in the back. Yeah, that's a good question. So in the beginning of the presentation, I talked about 3 stories that really inspired me when I was younger, which I know some people joined late, so I'll like go over them again. But yeah, my dad had, or he has post-polio syndrome. So that causes him to, like, one of his legs is like paralyzed, so he's not able to walk as a normal person. I was involved in volunteering at this like neuroplasticity physiotherapy clinic, which I met this war veteran who had a traumatic brain injury and wasn't able to walk. And then lastly, my grandpa, who unfortunately passed away this past summer due to Parkinson's. So my main motivation, I think, with interacting with these stories is to understand how the brain works and to see if there's ways in which we can solve like degeneration, disorder, and just anything that can go wrong in the brain. And I think initially going to the field, I was pretty set on like neurotech, which is kind of one of my first experiences that I had in high school. But I think I eventually got more interested in like exactly like how the brain works. So like not necessarily just like applying neuroscience and creating new technologies, but like more so applying like tools and data science and computation to understanding like specific mechanisms of the brain. So like memory, for example, like how different diseases arise. So more like the basic science stuff right now. Yeah.
What program are you in UW again?
I'm in biomedical engineering. Yeah.
Oh, that's really hard to get into.
I think it is one of the hardest programs in engineering with software. Yeah. I don't know. I think it's I think it's probably pretty competitive. I don't know how it is now, but are you in high school?
Yeah, it's like I'm not at all. So I think that's out of like the picture, which is really sad because I really like I'm really interested in biotech. So there are like an alternative, like options I can do like without physics.
I think a lot of my friends within this community have just like life sciences degrees, or they're in life science programs. And I think if you want to do a lot of this like more molecular, like cellular stuff, like that's a very good fit. For me, I didn't know I wanted to do this kind of stuff, so I wanted more like technical skills. But you can always like, learn like the technical skills like on the side, or have like a minor in like computer science or something. I also have some friends in like pure math, pure CS, and they have like bio like electives, and then they like integrate the 2 together. Yeah. Yes, in the blue hoodie.
How is bio?
Sorry, can you repeat that?
How does bio?
How does informatics, how does that fit into the field? Yeah, so bioinformatics, I would say like bioinformatics and computational biology, the terms are kind of used interchangeably. I would say like the main difference between bioinformatics and comp bio is that bioinformatics is like taking large collections of data and like figuring out how we can draw insights from that. Whereas like comp bio is more so like modeling and like simulating a certain system. So I would say like transcriptomics, that my technical example about transcriptomics is more like bioinformatics, whereas like the RNA folding example is more like comp bio, because that's like modeling and simulating a certain system. There's like different people saying, like there's like different definitions, but I would say like that's the main difference. Yeah.
Could you go more into more detail about your research at Strand?
Yes, some of the stuff I can't talk about because of confidentiality, but yeah, so I was looking into how, also by the way, if you guys need to leave, you can feel free to leave. I know it's like really over time, but I'll stay here for questions if you guys have more questions. So I was looking into how different RNA sequences bind to RNA binding proteins. So these proteins pretty much regulate like different functions in the body. And so if you're able to like figure out how this protein binds to RNA, that can tell you like how like basically how like the different mechanisms are regulated in the body. So I mean, this is pretty much the technical challenge I was given. Pretty much like how do you represent RNA sequences and extract patterns from it, and then predicting the final output, which is like whether there's like a binding protein, like or not. So yeah.
Like, were the proteins involved in like specific? You said that they were related to cancer, or?
So yeah, the company focused on cancer, but I didn't have that focus. So it was just like a general purpose like model. So these like binding proteins, they have a bunch of different functions that I cannot name right now. There's a lot, but yeah, different proteins kind of regulate different like bodily like processes and stuff. Yeah, there's a paper that I probably linked in the hack pack that is really related to what I worked on. I think it's this one. Yeah, I think this one is, yeah, binding preferences. Yeah, this one, if you want to look into that. Yes, the white hoodie.
You mentioned some system modeling things like, for example, the RNA strands and modeling using animal techniques. How do you come that to optimizing it for actual like problem solutions, such as like creating optimal like RNA vector definition?
So how do you go from the
Like system modeling to like optimization?
Yeah, so the question is if you can go, how can you go from modeling these RNAs, specific RNAs, right?
That's an example, yeah.
Yeah, to optimization. So I think the first part is modeling, like understanding which specific like, so in this example, like words, like subsequences of RNA bind to different subsequences. And so if we're able to see like, let me go back to my slides. Yeah, so if we're able to see like this word or this like k-mer is like most likely to bind to like this k-mer here, then we have like characterized like patterns within the RNA that kind of deal with like the specific function. And then how we go from there to optimizing is we can like test out how different, you can like replace those sequences with different words and see how like the function or the folding changes. And then this is like applied to drug discovery specifically because we want to like have really precise targets to like cancer cells, for example. So maybe like this one RNA that is not optimized targets like all cancer cells, whereas like one specific RNA strand that we did optimize targets like a specific subset of cancer cells. And that might be just like a small change in the sequence. That makes sense. Yeah. Okay, any other questions? Okay, this seems like we're all good. Feel free to, yeah, if any other questions come up. But thank you, and thanks for coming out.